Now imagine that you run a bookmaking factory and you've just printed up all the pages of your favorite book. Now that you have the pages, is the book ready to go? Well, books usually have front and back covers, so you might want to put these on. Also, where there are any blank or messed up pages made during printing, you should probably check for those and remove them before selling your books, or you might end up with some unhappy customers. The steps I just talked about are pretty similar to what happens to RNA transcripts in the cells of your body. In humans and other eukaryotes, a freshly made RNA transcript hot off the RNA polymerase process is not quite ready to go. Instead, it's called pre-mRNA and has to go through some processing steps to become a mature messenger RNA or mRNA that can be translated into a protein. These include addition of cap and tail molecules to the two ends of the transcript. These play a protective role, like a book's front and back covers, and removal of junk sequences called introns. Introns are sort of like blank or messed up pages made during a book printing, which have to be removed in order to for the book to be readable. So in this presentation, I will take a closer look at the cap, tail, and splicing modifications that eukaryotic RNA transcripts receive, seeing how they are carried out and why they are important for making sure we get the right protein from our RNA. So as a quick review, gene expression, the reading of a gene to make a protein or chunk of a protein, happens a little bit differently in bacteria and eukaryotes such as humans. In bacteria, RNA transcripts are ready to act as messenger mRNAs and get translated into proteins right away. In eukaryotes, things are a little more complex, though in a pretty interesting way. The molecule that's directly made by transcription in one of your eukaryotic cells is called a pre-mRNA, reflecting that it needs to go through a few more steps to become an actual messenger mRNA. So these are adding of a 5' cap to the beginning of the RNA, addition of polyp A tail of A nucleotides to the end of the RNA, and chopping out introns or junk sequences, pasting together of the remaining good sequences, which are exons. Once it's completed these steps, the RNA is a mature mRNA. It can travel out of the nucleus and be used to make a protein. So, what is this 5' cap and poly A tail? Both ends of a pre-mRNA are modified by the addition of chemical groups. The group at the beginning of 5' end is called a cap, while the group at the end, which is 3' end, is called the tail. Both the cap and the tail protect the transcript and help it get exported from the nucleus and translated on the ribosomes, which is protein making machines found in the cytosol. The 5' cap is added to the first nucleotide in the transcript during transcription. The cap is a modified guanine nucleotide and it protects the transcript from being broken down. It also helps the ribosome attach to the mRNA and start reading it to make a protein. How is the poly A tail added? The 3' end of the RNA forms in kind of a bizarre way when a sequence called a polyadenylation signal shows up in an RNA molecule during transcription. An enzyme chops the RNA into at that site, and another enzyme adds about 100 and 200 adenine nucleotides to the cut end, forming a poly A tail. The tail makes the transcript more stable and it helps it get exported from the nucleus to the cytosol. Okay, so next is RNA splicing. The third big RNA processing event that happens in your cells is RNA splicing. In RNA splicing, specific parts of the pre-mRNA called introns are recognized and removed by a protein and RNA complex called the spliceosome. Introns can be viewed as junk sequences that must be cut out so the good parts version of the RNA molecule can be assembled. What are the good parts? The pieces of the RNA that are not chopped out are called exons. The exons are pasted together by the spliceosome to make the final mature mRNA that is shipped out of the nucleus. A key point here is that it's only the exons of a gene that encode a protein. Not only do the introns not carry information to build a protein, they actually have to be removed in order for the mRNA to encode a protein with the right sequence. If the spliceosome fails to remove an intron, an mRNA with extra junk in it will be made and wrong protein will get produced translation. Okay, so alternative splicing. So why splice? We don't know for sure why splicing exists, and in some ways it seems like a wasteful system. However, splicing does allow for a process called alternative splicing, in which more than one mRNA can be made from the same gene. Through alternative splicing, we and other eukaryotes can sneakily encode more different proteins than we have genes in our DNA. In alternative splicing, one pre-mRNA may be spliced in either of two or sometimes many more than two different ways. For example, in the diagram you see, the same pre-mRNA can be spliced in 3 different ways depending on which exons are kept. This results in 3 different mature mRNAs, each of which translates into a protein with a different structure. So that's it, all about the eukaryotic pre-mRNA processing. So to summarize the key points, when an RNA transcript is first made in eukaryotic cell, it is considered as pre-mRNA and must be processed into a messenger mRNA. A 5' cap is added to the beginning of the RNA transcript, and a 3' poly A tail is added to the end. In splicing, some sections of the RNA transcript or introns are removed, and the remaining sections, exons, are stacked back together. And some genes can be alternatively spliced, leading to the production of different mature mRNA molecules from the same initial transcript.